So we'll move on to biochem. So biochem is mostly focused on a lot of the biology topics, which you if you've done biology before, it does make your life a lot easier in terms of the topics that you'll have to do. But if you haven't done bio, a lot of them will have the bio bridging course, and or you'll have it done during your undergraduate course, so you'll have some background knowledge going in. And stuff like this includes like cells and tissues. It includes protein transcription, translation, and kind of expression. It includes the ATP scale, includes microbiology and immunology as well. So let's just talk a bit about cells. I'm sure this diagram seems quite familiar to you. So if we just talk about the different things on the cells, we can be talking about, so this was like literally my first day, my first lecture. She showed us a cell, and asked us what all these different things were and what they did. And all I could think of was mitochondria is the powerhouse of the cell, because I was so tired from the holidays, and I just looked at the diagram and I was like, oh, this is giving me like flashbacks to biology and going through cells. So if we look at all of these different structures here, right? So we can see that that's the mitochondria, the powerhouse of the cell, which produces ATP, which is really important for us to have energy and to provide the energy in our cells for whatever we really need to be done. And it's also quite important because it does play a role in keeping our electrolytes, which is the salts in our blood, balanced. We'll also have these vesicles up here, which might be containing things, fun things, and we also might have lysosomes, which contain even more fun things, which degrade other things. If we look here, we're going to have the Golgi body here, which is like the post office of the cell, and that's what it packages. And then we have the smooth endoplasmic reticulum, which is important for lipid synthesis and lipid kind of packaging. And then we have the rough endoplasmic reticulum, which rough for all these ribosomes attached to it. And remember, that's what helps package the proteins once they're formed and helps actually form configurations. Otherwise, we just have long strands of random amino acids, or long strands of patterns which can't actually be folded together. And obviously, ribosomes are those little things which help you form where the proteins are. And then we have the wonderful nucleus and nucleoid, which so the nucleus is the outside area, and the nucleoid is the space which actually contains all the DNA and wonderful things that make you you. And essentially, that's where protein transcription will take place. So just keep that in mind. And essentially, it helps us do everything. It writes the script, it tells us what to do, right? Right now, every one of us is using our DNA to do various things, lots of wonderful miraculous things, right? Another thing with the DNA is it will have, so another thing about the nucleus and nucleoid, the DNA within that, the two strands of DNA, there are things which turn on and turn off, kind of like switches. And the switches within the thing, the combination of switches which are turned on and off will kind of form what the function of the cell is. So, for example, if this cell was a muscle cell and it helped you run, right? The thing that helps form all the mitochondria and helps it do that would be switched on, because they're saying we want lots of mitochondria so we can be powerhouses of the cell, and then we can power other things as well, so that we can run around, do what we need to, climb monkey bars, etc. Live our lives, climb things, be active, walk across campus, those types of things. You don't need to know about like plant cells. It's kind of irrelevant, just animal cells, because humans are animals. So that's why we just need to learn about animal cells, not plants. Types of tissue, this may be familiar to you, maybe not, but essentially the focus is on kind of there's four main types of tissue, and what we do is there's connective tissue, epithelial tissue, nervous tissue, and muscle tissue, right? Connective tissue, like the name suggests, connects different things together. And it can also include fat tissue as well, because that kind of forms strands between different things and holds them together, right? And some diseases affect connective tissues, right? So there's something called Ehlers-Danlos loss, ELHER hyphen DANLOS. not something you definitely, not something you need to know at this point. Something cool about it is it affects the connective tissues of us. And what it actually causes is because there's connective tissue everywhere in our body, it actually affects the heart and causes heartbeats to kind of balloon out and causes really bad problems. If you can imagine the artery, like our blood vessels kind of ballooning out, that's not a good thing, right? And another interesting thing is that it can cause like bluing of the white parts of your eye as well, and also cause hypermobility. So your joints and stuff can like flip over. People can dislocate their own hips, etc., etc. So quite a cool, oh, not a cool disease, but a very interesting disease, and it's all because there's a defect in the connective tissue. Another disease that is similar is something called Marfan syndrome, Marfan. And in that syndrome, once again, you get something, so it affects the connective tissue, so it really affects the big artery coming out of your heart, and it's pretty bad for that. And it can also cause issues with the gut as well. And it causes these people to be, on average, a bit taller than other people and to have really long joints as well. So very interesting things, and they're based on this type of tissue. Then we have epithelial tissue. Epithelial tissue is basically the tissue that just lines everything. So your epithelium is like the epithelium of the body is basically your skin, right? Because it lines your body. And there's something called endothelium, and your endothelium is the lining inside of your blood vessels. So that can also get damaged and cause problems as well. Then we have muscle tissue, and muscle tissue, there's a couple different types of muscle tissue. So there's smooth muscle tissue and skeletal muscle tissue. So smooth muscle tissue, the appearance of them are slightly different, but smooth muscle tissue is essentially controlled by our body's autonomic system. So the autonomic nervous system is essentially the system that doesn't require us to actively think and do. So it helps us breathe, it helps us go to the bathroom. It helps us see. It helps our blood pressure be stable. Just helps us do a lot of things that we don't actively think about or actively do, right? So that's control. That's called the autonomic nervous system. And it kind of makes sense because it's auto, right? And the two components of that is the sympathetic and parasympathetic. So you might think of the sympathetic as the fight or flight response, right? So when you see a bear charging towards you, that's your sympathetic nervous system being activated. So you get a fast heart rate that allows you to run faster. Um, you have your pupils dilating, which allows you to see better, etc. And then you have the parasympathetic, which is the one which is rest and digest. That's the other name for it. So you're sitting, you're eating, you're chilling. That's your parasympathetic system. Cool. Okay. And the other one is your skeletal muscle, which is controlled by your somatic system. So somatic is basically the system which you actively need to do. So when I'm gesturing, moving my hands, moving around, I'm all using my skeletal muscle. And because even though I do not actively think I want to move my hand, when I move my hand, a signal goes to my brain to say, okay, she's moving her hand now, and that goes to my brain. And then I sense I want to move, right? So I say, okay, let's say I want to move my arm, it goes, the signal goes to my brain and then comes back down. Compared to an autonomic thing, like I do not actively have to initiate the movement, it will just happen inside of me. Um, cool. Okay, so those are the different types of muscle. And the heart muscle is a type of skeletal muscle, but it's a little bit different in terms of the appearance of it. But you would think that the heart muscle is completely made of smooth muscle, completely made of skeletal muscle. It's a little bit of a hybrid of both and it doesn't really look like either of them. But it actually is a skeletal muscle, and essentially what it is it has like an activation system, which is like a kind of auto loop activation. Okay. And then nervous tissue is your neurons, the things that help you think, help you move, help you do things, the things which connect your hands to your brain to say, okay, I'm moving my arm now. Cool. All right. So in terms of transcription and translation, there's a process called nuclear expression, right? So I would think that this is something also that you'll probably just memorize and learn at some point, but just to go through the process, we have And this will also be useful if you're doing any bio-related subjects in general. But essentially, we have this first step here, right? And this is where we have this unlooping of these two strains, right? The strains break apart and kind of unfurl because they were wound together before, right? So this RNA polymerase, right? This RNA polymerase, some type of thing here, will bind to this section, bind to a section in the DNA, and it will help this unfurling, and transcription factors will come along and be helpful little elves and help it unwind as well. So then what it does, the RNA polymerase binds the promoter region, which is the region which is saying, hi, hi, come bind here. And then it moves along the template strand and it reads it from a 3 to 5 direction. Now, one side of the thing is 3 and one side of the amino acid is 5. I would say you don't necessarily have to know it very, very in detail, but just knowing that it it can go from 3 to 5, and it's named that way essentially because we need to name it so that it has a 3 and 5 cap end, so we can know which end to put which things on, right? So it does it in an anti-parallel fashion. So it reads it from 3 to 5, but it makes the strand from 5 to 3. All you have to remember from that sentence is essentially that it binds to it and it forms a complementary chain. So it just does the opposite. And you will have heard something like ATCG, so adenosine, tyrosine, and then cytosine, guanine, right? So those kind of amino these bases bind together and they just paired up depending on which one it is, and you just need to find its pair essentially. So then it moves until it reaches the end of the gene. And at that point, there'll be a termination sequence being like, stop, stop, stop. And then it will release something called a pre-RNA. Now, I thought pre-RNA was the full name of what it was. It's actually something called a preliminary mRNA. But essentially, this preliminary mRNA will be complementary to the template strand that's formed. And it'll be the exact same as this coding strand, which is the opposite strand to the template strand, except the T and U will differ depending on whether it's RNA or DNA. And then what will happen after this, before it moves to out of the cell into the translational phase, is we'll have introns sliced out. So introns and exons are essentially just segments in the DNA. So the introns are the things which are included, exons, sorry, are the ones which are extra and that are just taken out, right? And then we have this, so if you can imagine like segments and then taking out, it now becomes a lot shorter. Now, remember that 5N and 3N we were talking about before? So we add a cap to the 5N, so we think of the 5N as like the head, so we add a cap to it and add a little tail of poly A. So that's literally A. That's what a poly A tail is to the other end, so to the tail end, right? Okay, so then once this is made, now we have a mature mRNA which can move out of the cell. And this mature mRNA will then go to this translational cell. And this translational cell essentially just helps us find out what's going on. So it it reads this mRNA, right? And then it attaches to those ribosomes which were on the rough endoplasmic reticulum from before, and it will start reading at the 5N, and it will start reading, and once it gets to AUG, the start codon, it will start reading and pumping out this protein chain. The really important thing is the start. You don't need to memorize the start codon, definitely not, but essentially you just need to know that there's a certain code which starts the production. As it reads, so a codon is 3 letters in a DNA sequence or an mRNA sequence. So it's like AUG or AGC or whatever, right? Like it's 3 letters, and those 3 letters, there will be something called a tRNA, right? And this tRNA will come over like a little fish, and if it matches that mRNA sequence, it will attach on and then release its amino acid, right? And it does that every single time. And when the amino acids are together, what they get done is they bond together. And by that way, we're kind of forming a chain of amino acids. So We get the tRNA, it forms a bond with the side, it releases, it lets go, and it also lets go of the mRNA as well. So it's not a permanent binding, it's very temporary. So we can kind of see it being done here. So you see how it kind of comes along, the tRNA, oopsies, the tRNA comes along, it binds, it leaves, it binds, it leaves, it binds, it leaves, it binds, it leaves, etc., etc., right? Cool. So this goes on and on and on. And by the way, that little segment on the tRNA is called the anticodon. On the mRNA, it's called a codon, and in the DNA, it's called a triplet. Okay. So then it gets coded, coded, coded until we reach a stop codon. So a sequence of DNA where it's like stop, 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 right? And once we get to that, this polypeptide chain, which is that chain of amino acids which are being bonded together, will now just break off. And it will be released into the wild. And once it's released into the wild, then it will go to the rough endoplasmic reticulum and get folded and packaged, and then might get sent out of the cell, used within the cell, and etc.